You know, when we analyze a medical diagnosis, there's usually this, like, underlying expectation of anatomical precision.
Oh, absolutely.
Right. Like, you look at an X-ray of a fractured radius and you see that jagged white line, and the pathology is just undeniable. It's binary. It's broken or it's not broken.
Yeah, and I mean, it's a very comforting framework. We are totally wired to categorize pathology into those neat, observable states.
But the moment you step into the landscape of oncology specifically, the evolution of sarcomas, that rigid diagnostic framework just completely collapses.
It really does. It falls apart.
Yeah. We're no longer looking at a static injury. We're looking at a system that literally shapeshifts over time. So today we're bringing you along on a deep dive into an incredibly dense mechanistic study. This was published recently in Nature Communications on June 17th, 2026.
A fascinating paper.
It is. And our mission today is to examine how a team of researchers essentially reverse engineered a highly aggressive cancer.
Yeah.
They went sequential mutation by sequential mutation to basically uncover its therapeutic kill switch.
Right, and the specific pathology we're unpacking today is Neurofibromatosis Type 1, or NF1.
Yeah.
So for those of you familiar with hereditary cancer syndromes, you probably know that NF1 patients carry a genetic predisposition to develop these peripheral nerve tumors. They're called plexiform neurofibromas, or PNFs.
And usually those stay benign, right?
Exactly. In the vast majority of cases, they remain benign, though they can still be clinically complicated. But in roughly 10 to 15% of patients, we see this devastating malignant transformation. The benign PNF progresses into a malignant peripheral nerve sheath tumor, or MPNST.
Which just completely changes the clinical reality for the patient.
Completely.
Because MPNSTs are exceptionally aggressive soft tissue sarcomas. They just entangle themselves in the peripheral nervous system. And systemic therapies have historically fallen remarkably short, you know.
Yeah, outside of early aggressive surgical resection, the 5-year survival metrics are grim. They really are.
So what's the holdup? Why has it been so hard to develop targeted therapies for this?
Well, the historical challenge is that we typically only get to study the end-stage tumor.
Right.
By the time it's resected and sequenced, the cancer's genome is just an absolute chaotic mess of passenger mutations and chromosomal instability.
It's a ton of noise.
Exactly. So working backward from that end-stage pathology to find the actual driver mutations is incredibly difficult.
It's basically the classic problem of arriving at the scene of a completely burned down building, right?
Yeah.
And you're trying to determine which specific spark ignited the fire, but you have to ignore all the secondary explosions that happened after.
That's a perfect way to put it, which brings us to the brilliance of this specific study's methodology.
Right.
Instead of sifting through the ashes of an end-stage sarcoma, these researchers engineered the fire from scratch.
Which is wild.
It is. They utilized human-induced pluripotent stem cells, iPSCs, and CRISPR-Cas9 gene editing to chronologically build the tumor in the lab. They enforced the exact biological sequence of the cancer's presumed evolution.
So by constructing this progression artificially, they gained the ability to, like, pause the oncogenesis at each distinct evolutionary stage.
Exactly. They could map the chromatin landscape, they could observe the transcriptome, and most importantly, identify transient synthetic lethalities.
Vulnerabilities that only exist during specific developmental windows.
Spot on.
Okay, so if we are mapping out this evolutionary blueprint, we have to start at the genetic cascade itself. How does a structurally sound neural crest cell devolve into a lethal sarcoma? Like, what is the recipe here?
So the researchers validated a very strict multi-hit genetic cascade. The initiation event step 1 is the biallelic loss of the NF1 gene itself in a neural crest lineage.
Okay.
When that tumor suppressor is lost, the cell begins to hyperproliferate, and that forms that benign plexiform neurofibroma. In the context of their model, they call this the 1KO, or single knockout stage.
So the benign mass is established. But to advance toward malignancy, the cell requires a secondary structural failure.
Right. That secondary failure is the deletion of the CDKN2A locus. When the tumor loses this specific chromosomal region, it undergoes this histological and biological shift into a pre-malignant lesion.
Clinically categorized as an ANNUBP, right?
Yes, an atypical neurofibromatous neoplasm of uncertain biological potential. I know, it's a mouthful. But this creates the researchers' 2KO, or double knockout model.
Now, looking closely at that CDKN2A locus, this isn't just like a simple single-function gene deletion.
Far from it. The CDKN2A locus is fascinating because it utilizes alternative reading frames to encode two entirely distinct tumor suppressor proteins.
Okay, what are they?
P14ARF and P16INK4A. Both of these proteins are critical regulators of the cell cycle. They essentially monitor cellular stress and DNA damage. If the cell begins replicating dangerously, they trigger cell cycle arrest or apoptosis.
So it's basically a redundant dual braking system in a semi-truck. You've got the P14 brake line and the P16 brake line.
Yep.
But looking at the mechanics of this, I have to push back a bit. If the goal of the cancer is just to bypass cell cycle arrest, wouldn't severing just one of those brake lines be sufficient? Like, why does the pathology require the deletion of the entire locus?
That is a phenomenal mechanistic question, and it's actually one the researchers explicitly tested.
Oh, really?
Yeah. They used their CRISPR platform to generate two variations of the 2KO cells. In one cohort, they selectively knocked out only P16, so they left the P14 reading frame fully functional.
Yeah.
And then in the second cohort, they deleted the entire locus, eliminating both P14 and P16.
Right, so completely cutting the brakes.
Right. And to observe the physiological impact, they took these engineered neural crest spheres and performed orthotopic engraftments directly into the sciatic nerves of immunocompromised mice.
Wow, placing them directly into the native peripheral nerve microenvironment. What did the pathology show?
The divergence was stark. The cohort that lost only P16, so that secondary P14 braking mechanism was still active, they struggled to establish any meaningful pathology. The outgrowths were minimal and histologically benign.
Interesting.
But the cells lacking both P14 and P16, they rapidly developed robust, highly cellular tumors that perfectly mirrored human atypical neurofibromas. So the physiological environment actually requires the complete catastrophic failure of both tumor suppressors for the cancer to advance.
Which tells us the cell is under immense oncogenic stress, and any remaining functional tumor suppressor will just force it into senescence.
Exactly.
And establishing this rigorous 2KO model allowed the researchers to identify a major metabolic consequence, right? Because when you cut both brake lines, the cell doesn't just divide, it has to physically synthesize enough genetic material to support that rapid division.
Yes, that metabolic consequence is exactly what they found when they ran RNA sequencing on these 2KO cells. Compared to the benign 1KO stage, the double knockout cells showed a massive upregulation of a gene called TYMS. Right. TYMS encodes for thymidylate synthase, which is an enzyme absolutely critical for generating DTMP. And DTMP is a fundamental nucleotide precursor required for DNA synthesis.
So the cell is basically desperately hoarding the building blocks of DNA because it's trapped in this state of hyperproliferation. And that dependency creates a massive vulnerability.
It creates a classic synthetic lethality. Because these pre-malignant cells are so heavily reliant on thymidylate synthase just to survive their own replication cycle, the researchers targeted them with a drug called pemetrexed.
Okay.
It's an antifolate drug that directly inhibits that specific enzyme.
So without that enzyme, the cancer cells essentially starve themselves of nucleotides right in the middle of trying to replicate their genome.
Precisely. The inhibition starves them of DTMP, and that leads to catastrophic replication stress, uracil misincorporation, and DNA double-strand breaks. Morphologically, they actually observed the Pemetrex-treated cells developing these massive aberrant nuclei before undergoing apoptosis.
Oh.
The corrupted replication machinery essentially jammed and destroyed the cell from the inside.
That is incredible. Okay, let's consolidate this conceptually for you listening, because the chronological precision here is so vital. We're going to do a quick recap.
Go for it.
So far, we have a two-step evolutionary process. Bullet 1, the NF1 loss initiates the benign proliferation. Bullet 2, the total loss of the CDKN2A locus disabling, both the P14 and P16 breaks, drives the tumor into a pre-malignant state, which simultaneously exposes a critical dependency on nucleotide synthesis.
Perfect summary.
And think about how precise this is. Here's a question for you listening. What stands out to you about how the cancer has to perfectly dismantle the cell's defenses just to survive? Because capturing a tumor in this transient 2KO window could theoretically allow us to leverage drugs like Pemetrex before the sarcoma ever fully forms.
And that is the ultimate prophylactic goal. But, as we know, biology rarely stalls at the pre-malignant stage. We have to look at the transition to true malignancy.
Right. Because despite all this aggressive replication, an NEBP is still technically pre-malignant. It lacks the invasive, shape-shifting characteristics of a true sarcoma. There's an evolutionary gap. So what's the mutation that actually pushes this mass over the edge into a full-blown MPNST?
The final driver is the disruption of the polycomb repressive complex 2, or PRC2. In clinical MPNST genomics, we frequently see mutations in a gene called SUZ12, which is a core structural subunit of PRC2.
Okay.
By utilizing CRISPR to knock out SUZ12 in their model, the researchers generated the ultimate 3KO, the triple knockout cell line.
NF1, then CDKN2A, then PRC2.
Yeah.
But looking at the sheer genetic instability of these cells, mutations happen randomly all the time. Did the researchers find that these specific knockouts could occur in any order?
This is where the study uncovered a massive biological constraint. The sequence is entirely non-negotiable.
Really?
Yeah. The researchers actually attempted to induce the PRC2 knockout in cells that still possess functional P14 or P16 genes. So essentially, they tried to skip step 2 and force the tumor straight from benign to malignant.
And the microenvironment rejected it.
The cells rejected it autonomously. When PRC2 is lost, it creates this enormous wave of epigenetic dysregulation. If the cell's tumor suppressor breaks P14 and P16 are still intact when that dysregulation hits, those proteins sense the chaotic stress and instantly trigger irreversible cellular senescence.
Oh, wow.
The cell essentially locks its own genome down permanently to prevent malignancy. The cancer absolutely must eliminate CDKN2A first, completely blinding the cell to its own dysregulation before it can survive the loss of PRC2.
I love the mechanics of this. It operates exactly like a strict combination lock. Like, imagine a 3-digit padlock for you listening. You cannot turn dial 3 PRC2 until dial 1 NF1 and dial 2 CDKN2A are perfectly aligned and clicked into place.
Yeah, that's exactly.
If you attempt to force the third dial prematurely, the internal stress mechanisms jam the lock permanently and the tumor dies.
That combination lock analogy perfectly captures the evolutionary pressure the cancer is under. And to prove that this specific 3-part combination was the true driver of the sarcoma, they subjected the 3KO cells to the definitive in vivo assay.
Back to the mice.
Back to the mice. They took these lab-grown, triple knockout neural crest spheres and engrafted them into the sciatic nerves of their mouse models.
Now, orthotopic engraftment is notoriously challenging, right? Because you have to wait to see if the cells can actually survive and adapt to the host's nervous system.
It requires immense patience. But 4 months post-engraftment, the pathology was undeniable. The mice developed a large, aggressive, highly cellular tumors at the injection sites. These masses were packed with mitotic figures and histologically mirrored early-stage human MPNSTs.
But wait, how do we ensure we aren't just observing a bizarre reaction from the host mouse's own biology? Like, how do we definitively prove those are the exact human cells engineered in the lab?
Through immunohistochemistry, the researchers stained the tumor sections for Q80.
Q80? What is that?
It's a nuclear protein exclusively expressed in human cells. The malignant tissues lit up, confirming definitively that these aggressive sarcoma cells were the direct descendants of the engineered human iPSCs. They had successfully built an MPNST from scratch.
That is just phenomenal, but it forces us to ask the ultimate mechanistic question. We know what genes were deleted to trigger this, but how does the loss of PRC2, that final dial in the combination lock, physically transform a localized mass into an invasive sarcoma? Like, what is the actual mechanism?
To answer that, we have to transition from genetics to epigenetics. PRC2 isn't a traditional gene that codes for a structural protein. It's an epigenetic remodeler.
Okay.
Its primary function is to lay down specific methylation marks, specifically H3K27E3 on the histones that package our DNA.
And those methylation marks act as silencing signals, right?
For an audience familiar with chromatin accessibility, we know that if chromatin is tightly condensed by these polycomb-mediated repression marks, the transcriptional machinery just cannot access those genes. The genes are physically locked away. Yes. PRC2's job is to maintain the cellular identity by keeping certain genomic regions tightly repressed. When the SUZ12 mutation destroys PRC2, those repressive H3K27Me3 marks vanish. The entire epigenetic landscape of the cell physically shapeshifts.
Wow.
And the researchers utilized ATAC-seq, which maps chromatin accessibility, to observe this global reorganization in real time.
What did the ATAC-seq data reveal about the cell's identity crisis?
It revealed a dual mechanism of catastrophe. First, the chromatin paradoxically closes and condenses around a master transcription factor called SOX10. Now, SOX10 is absolutely vital for normal gliogenesis. It's the molecular beacon that tells the cell to remain a stable peripheral Schwann cell.
So by shutting down the SOX10 promoter, The cell permanently loses its glial identity. It literally forgets what it was supposed to be.
That's the first half. The second half is the sudden catastrophic opening of chromatin across entirely different genomic loci. Regions that should be tightly repressed in a neural cell suddenly lose their methylation marks and unwind.
So chaos ensues.
Absolute chaos. The researchers saw massive activation of a neuromesenchymal transcriptional program. This was characterized by the overexpression of genes like SOX9, TWIST1, and PRRX1.
And those are genes classically associated with primitive mesenchymal stem cells, right? Traits that drive invasion, migration, and aggressive survival.
Exactly.
If we visualize this transition, let's unpack this for the listener. It's essentially a catastrophic rerouting of cellular fate. Like a train switching tracks, you can imagine the cell traveling down a highly regulated track toward, let's call it, normal glial city.
I like that.
Suddenly, the tracks leading there, the SOX10 bridge, are destroyed, halting that forward progress. But simultaneously, a dormant primitive track opens up, and the cell is violently diverted onto the mesenchymal express. The cell devolves from a specialized nerve component into a primitive invasive entity.
Yes. In the oncology literature, this is known as the glial-to-mesenchymal transition, or GMT. And the ATAC-seq data provided conclusive proof that this GMT, driven entirely by the epigenetic shockwave of losing PRC2, is the fundamental hallmark of MPNST pathogenesis.
Let's do another quick recap of this epigenetic shift for you listening, because this is dense.
Sure. Bullet 1. PRC2 loss is the ultimate trigger. Bullet 2. It closes chromatin for healthy development, silencing SOX10. Bullet 3. It opens chromatin for primitive, aggressive traits, activating SOX9.
And here's a question to ponder. When you picture the DNA literally spooling tighter to hide healthy genes, how does that change your view of what cancer really is? It's not just a collection of broken genes, it's the total reprogramming of the cellular architecture.
It really is a systemic architectural hijacking.
Yeah.
And understanding that architecture is exactly what allowed the researchers to finally hunt for a cure.
Which brings us to the translational climax of the study. We have a biologically accurate, highly characterized 3KO model of early-stage MPNST. Now, how do we kill it? How do we exploit this chaotic new architecture?
The researchers transitioned into high-throughput screening, but they made a crucial methodological upgrade. Instead of screening drugs against a two-dimensional monolayer of cells in a flat petri dish, they cultured their 3KO cells into three-dimensional spheroids.
Which is vital for translational relevance. We know that traditional 2D cultures often completely fail to predict real human tumor responses. They lack hypoxic cores, they lack natural nutrient gradients, and realistic cell-to-cell adhesion mechanics. Using 3D spheres acts like real microtumors, ensuring the epigenetic state actually mimics human pathology.
Exactly. And because the fundamental driver of this cancer is that massive epigenetic dysregulation we just discussed, the researchers didn't just throw standard chemotherapies at it. They specifically screened the spheroids against the MPC epigenetics library.
What's that?
It's a highly curated collection of 339 small molecule compounds designed specifically to target epigenetic modulators.
So they were looking for a compound that would uniquely weaponize the cell's unwound chaotic chromatin against itself. Out of those 339 compounds, what actually worked? What demonstrated selectivity against the 3KO model?
The screening data heavily highlighted PRP inhibitors, specifically olaparib. The 3KO spheroids were profoundly sensitive to PRP inhibition compared to the earlier benign stages.
Now, that is a fascinating therapeutic intersection because PRP proteins are primarily known for their role in the DNA damage response, repairing single-strand breaks. Why would a tumor driven by an epigenetic mutation suddenly become exquisitely dependent on a DNA repair mechanism?
It comes down to replication stress. Because the loss of TRC2 unwinds the chromatin and forces the cell into that aggressive mesenchymal transition, the genome becomes wildly unstable.
Right.
The cancer is replicating at immense speeds with a completely disorganized architectural blueprint. To avoid total mitotic catastrophe, literally tearing its own DNA apart during division, it relies heavily on PRP to constantly patch up the microtears in its genome.
Oh, wow. So by introducing olaparib, you're taking away the only duct tape holding this chaotic mesenchymal genome together.
Exactly. It induces synthetic lethality by exploiting the tumor's own aggressive biology. However, and there's always a however, treating aggressive sarcomas with monotherapy is clinically precarious. MPNSTs are notorious for developing acquired resistance mechanisms and mutating around single-agent therapies.
which means we need a synergistic combination. We need to hit the DNA repair, but we also need to shut down the oncogenic signaling driving the proliferation in the first place.
The researchers deduced the exact same thing. They hypothesized that combining olaparib with an MEK inhibitor could provide that grand finale synergistic knockout. They selected selumetinib, a potent MEK inhibitor that aggressively suppresses the MAPK signaling pathway.
And that's a pathway hyperactivated by that initial NF1 loss, right?
Yes. And to validate this golden combo, they moved to the absolute gold standard of preclinical testing, the PDO-X model.
The patient-derived orthotopic xenograft.
Right. They took actual malignant tissue derived from a human MPNST patient and implanted it directly into the sciatic nerve of a mouse. This is as close to treating a human patient as you can possibly get in a preclinical setting.
How did the tumor respond to the dual blockade of olaparib and selumetinib?
The synergy was remarkable. The combination therapy was well tolerated by the murine models and induced significant suppression of tumor growth in vivo. It vastly outperformed either drug as a monotherapy. By simultaneously crippling the tumor's ability to repair its chaotic DNA with olaparib and suffocating its proliferative signaling with selumetinib, they essentially backed the sarcoma into a metabolic and structural corner it just could not escape.
It is a masterclass in targeted rational design. Zooming out and looking at the big picture of this Nature Communications paper, what is the ultimate takeaway for the future of oncology?
The overarching triumph here is really methodological. This study proves that we're no longer limited to analyzing the static terminal stages of disease. By leveraging iPSC-derived models and CRISPR, we can flawlessly map a cancer's step-by-step evolution.
Right, bridging the gap between flat lab cultures and live organisms.
Exactly. We can map the specific epigenetic transitions, build physiologically accurate 3D models, and accelerate the discovery of combo therapies for devastating rare diseases that currently lack standard of care options.
It is effectively a molecular time machine. We can finally watch the bomb being built, rather than just analyzing the crater. Which leaves me with one final provocative thought for you listening to mull over on your own.
Let's hear it.
We learned today that this specific deadly cancer relies on a strict password sequence to survive. First NF1, then the total loss of CDKN2A, and finally PRC2. We know that if that exact order is altered, the cell triggers its own senescence. So as we advance our capabilities in gene editing and epigenetic modulation, what happens in the future if we develop therapies that artificially scramble that sequence in high-risk patients before the tumor even forms?
Oh, that's interesting.
Right. If we detect a benign NF1 mass, could we theoretically force a premature epigenetic shift, intentionally jamming the cell's combination lock and forcing the cancer to trigger its own self-destruct sequence?
It fundamentally shifts the paradigm, from trying to kill the cancer to tricking the cancer into killing itself based on its own biological rules. It's an incredibly promising frontier.
It certainly is. Thank you for joining us on this deep dive into the architecture of oncology. Keep questioning the hidden mechanisms around you, keep looking for those evolutionary vulnerabilities, and we will catch you next time.